This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 58, recorded on June 13th, 2013. Hi everybody, I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, how you doing?
I'm well, and you? Back in San Diego?
Yeah, fine. After the ASM meeting, it takes me about a week to recover between all the things that went on there and the things that went on in my absence here. It takes a while, doesn't it?
Well, after a few days after Denver, I went up to Edmonton for a 2-day meeting, and then I just came back from Seattle for a study section. So now I'm going to recover.
Right.
And study section is more than a full-time job. It's hard work.
That voice that you hear is none other than Michael Schmidt from the Medical University of South Carolina. Hello, Michael.
Hello, Vincent. Hello, Elio. How is everyone today?
I just finished a 4-year stint on study section, so I am finished.
Oh, wow.
And I had already done 7 years previously, so 11 years. That is it. No more for me. I'm stating it now. No ad hoc, nothing. I'm finished. I need that time back. Think that's all right?
Thank you for your service. Those of us throwing proposals over the fence and lighting the candle in the cathedral, thank you.
Well, now I have to write some proposals. I just got out of a faculty meeting, and our chairman told us to write more grants.
No kidding.
That's the first item on our faculty meetings agenda every month. How many proposals have you submitted this month?
Yeah. Well, he used Arturo Casadeval as an example. He said he sends one in every cycle, or at least he used to.
But Arturo never sleeps.
Is that right?
I don't think a man sleeps, to tell you the truth. He is very prolific in his writing and his interaction with students, and he's on the speakers. He plans the general meeting.
MBio editor-in-chief. Right?
MBio editor-in-chief. I don't think the man sleeps.
Oh, I'm getting tired.
So we thank him for his service as well.
Oh, I'd say.
Well, we're back home, but Michael, you're not home.
No, I'm up in Washington, D.C. at ASM headquarters, and thanks to Chris Kondayan and Ray Ortega, they're letting me use the master studio of TWiM, so I may actually sound better today.
It's nice there, isn't it? It's Chris's office. It's really nice.
Oh, yeah. It's I could move in. He's got windows and everything.
Yep. And the rest of ASM there, you must feel erudite, right?
That's true.
All right. Today we have 2 very cool papers for you. As usual, always something cool here from TWiM. And the first is in a journal that I'm not sure, I might have read a paper or 2 in previously, but I know we have not done one on TWiM, right, Elio? Ecology Letters?
Certainly not my usual reading.
Do you scan this journal for articles, or you just stumble across this one?
Actually, I came across this. I don't. I came across this from one of the graduate students in the course I just finished teaching.
Oh, cool.
Who knows that I would dig a subject like this. So he sent it to me, and I was eternally grateful. And this paper may make it into Small Things Considered yet.
Oh, I would guess it would, yeah.
Is his backup career plans writing screenplays for Hollywood? Because this paper, this is a spoiler alert. I'm going to spoil it for folks. This paper is literally the basis of the script for Avatar.
Just about.
The movie, not the cartoon.
No, the movie, not the cartoon. That is correct.
Okay. So, should I tell you the title?
Please do.
It's Underground Signals Carried Through Common Mycelial Networks Warn Neighboring Plants of Aphid Attack. Okay? And it's by a bunch of folks in Northern England and Scotland, Aberdeen and the Rotham's station. The title tells a story. I mean, often good titles do. This one has pizzazz in addition to everything else, doesn't it?
Mm-hmm.
Yes.
Underground signals. Boy, that sounds like Michael was talking about. It sounds like a movie in the making. Carried through common mycelial networks, and they warn neighboring plants of aphid attacks. Now, let me tell you what this is about. It's about mycorrhiza, the very famous symbiosis that unite plants with their environment via fungi, fungal mycelium. So as you learned in high school, most plants, perhaps 80% of all plants, have a connection with fungi underground. These mycorrhizal fungi provide something you could call something like drinking straws. They extend the roots of the trees over a vast area, sop up water and nutrients like phosphorus and things like that, and carry them to the plant root. The plant root pays back by supplying the fungi with sugars they need. Remember, fungi are not autotrophic and plants are, so this is a good swap. So mycoplasma mycorrhiza. Oh boy.
It's okay. We got you.
Mycorrhiza constitute a huge network underground.
How huge?
Well, in terms of size, I once read a paper and I couldn't find it. I tried my darndest to find it, and if some of the listeners can put me onto it, I'd like to hear about it. Somebody proposed that the whole Earth, all the soil in the Earth, is connected by one single network. That's a little big. But if you don't believe that it's got to be that big, and besides, how do they cross the oceans? But anyhow, if you don't believe that, you'll believe the following, that there are places where there are huge accumulation of mycelium underground. There's a place in Oregon which is about 10 square kilometers in size. And the way you know it is because the mushrooms that are the fruiting bodies of this particular fungus show up over that area. And if you analyze them, you find out that the same organism, a single organism over 10 kilometers square. And by extrapolation, this may be one of the oldest organisms on Earth. It may be something like 2,000 years old. Anyhow, so that's that.
And in fact, they have a humongous fungus festival in Michigan. And the mushroom that they have associated with that weighs roughly 11 tons, or roughly 600 pounds per acre, and occupies 37 acres.
Right. There are such things are known, and they're probably an underestimate of how big this can get. It can get to be really, really huge. In addition, mycorrhiza are really important. Without plants which are reared aseptically or quasi-aseptically without mycorrhiza don't do as well. And I'm talking about most crops, most rainforests, most grasslands, and especially when places which are poor in phosphorus, the presence of mycorrhiza makes a huge difference. In fact, when people restore forests, that's the term for what you do after you destroy the forest. If you reseed the forest with seedlings, you make sure that you add mycorrhizal fungi to it. It can be bought commercially. There's one called Pisolithus tinctorius, which is, I think, widely used in reforestation. And if you don't do that, the trees don't do as well. So we have mycorrhizal fungi to thank for tremendous amount of what goes on in the world, and just amazing. In addition, and this is kind of funny, they can be seen in fossils, which are from the time that plants came to land from the ocean, which is something like 400 million years ago. Mm-hmm. And so if you section those, if you look at sections of these fossils, you find fungi from the very beginning. Some are inside the plant, some are on the outside of the plant. And in fact, mycorrhiza come in two kinds. They are the ectomycorrhiza, which means that they are found on the outside of the plant, of the root, and they are the vesicular arbuscular mycorrhiza. Forgive my complicated terms. These refer to fungi, mycorrhizal fungi that do penetrate into the plant root, and they make structures called vesicles and structures called arbuscles. Arbuscles, that's arbuscles, means small trees, because they look like they're really branched structures. And they are within the plant cells. So they can do their job either as a mantle around the roots or inside. And there are different kinds. The ectomycorrhizal comprise many of the mushrooms, just like we were talking. The mushroom we were talking about that Michael mentioned from Michigan and I mentioned from Oregon is called the honey mushroom, Armillaria, different species of Armillaria, which, by the way, is pretty good eating. Goes very well with pasta.
But to really put this into perspective, what you're describing is, can be reduced to a simple question: how do you make dirt? And soil carbon is so important to the global ecosystem. And in fact, it's the research that's out there today shows that the mycorrhizal fungi hold 50 to 70% of the total carbon stored in leaf litter on forest islands. And the paper I was reading, it happened to turn out for Sweden. But when you begin to think about the whole issue of climate change and CO2 in the atmosphere and the importance of carbon fixation and carbon sequestration, this is when you use sequestration in a good way of being able to capture that carbon. The mycorrhiza are absolutely critical to this process, and I think this is why the graduate student threw this paper over the transom to you, because as we're going to see, the signaling that these things are doing elevates the importance of mycorrhiza to even a more important level in terms of protecting the plants which feed the mycorrhiza that facilitate the carbon storage.
Since you brought this up, let me add one more thing, something that I actually just ran into not long ago. Mycorrhizal fungi make a protein called glomalin. You can find that in Small Things Considered. Glomalin is a very strange molecule that makes, as tough as can be. It lasts in soils for maybe something like 35 years, and to study it, you have to use very strong reagents. Anyhow, what it does, it binds soil particles together. So it not only constitutes just carbon, and by the way, this is about 30% of all the carbon in soil. Not only does it represent a lot of carbon, but it also changes the physical and mechanical properties of soil for the better. So fungi do a lot of things with the plants directly and with the plants indirectly.
I have a feeling that they are not very well respected, are they?
Well, when you say mycorrhiza, you get a yawn from most people. And in reality, they are essential for our life on Earth. I mean, we could live without them, but our crops would be maybe a third the size, something like that. I haven't seen a good estimate, but my guess is that without mycorrhiza, we would get a third the potatoes and a third the corn that we have, something of that sort.
Especially corn, because the nitrogen is only being left behind from the mycorrhiza. Corn doesn't fix nitrogen like beans do.
Right. So anyhow, it's big time. So these authors went and thought, what else do mycorrhiza do? And they knew that they are involved in signaling between plants because they can tell that plants with mycorrhiza talk to their neighbors. For instance, marigolds, through their mycorrhiza, inhibit their neighbors. In other words, they don't want to have many more plants around using their resources. So they send out a signal that inhibits the neighbors. And tomato plants do something very interesting. when they're connected via mycorrhiza with plants which have an early blight, they themselves become resistant. So there are every reason to believe that the plants talk to each other via this intricate meshwork that's on the ground. By the way, mycorrhiza are fairly non-specific. That is, the same fungus will make mycorrhizal relationship with a large number of plants. And this is important here.
So I guess there are many different species of these mycorrhiza, right?
Yeah, well, the ectomycorrhiza, the ones that don't penetrate, are an awful lot of fungi. A lot of mushrooms are ectomycorrhizal, so there are probably thousands and thousands of those. The endomycorrhizals, for those of you who remember your mycology, are part of the zygomycetes, like mucor and rhizopus, a different group than the ascomycetes, which make up most of the fungi on Earth, and which include most molds and yeasts and so forth, or the basidiomycetes, which is what we're just talking about, which is what most mushrooms are made of. These are zygomycetes, which are soil organisms. They include some human pathogens and so forth. They're common, but I don't think that the number of species of these vesicular or vascular mycorrhiza is that great. They're probably in the hundreds, not in the thousands. By the way, the plants like it so much that they make a chemotactic factor which attracts the fungi. This is a terpenoid lactone called strigolactones, for which there are many that derive from carotenoids, and they're secreted by the plants, and the fungi know where they are, and off they go looking for their maker, for the plant that made them. Mycorrhiza pay back in yet another way, which blew my mind. They make factors which increase nodulation by bacteria. Nodulation, as Michael just alluded to, is how all legumes get their nitrogen, and a huge source of nitrogen fixation on Earth. Huge. The principal one, nitrogen, making ammonia is something that nitrogen-fixing bacteria make. Anyhow, nodulation is good for legumes, and mycorrhizal plants help in nodulation. So now, let's come to the question of the paper. If mycorrhizal are a conduit of signal between plants, might they not be involved in signaling resistance to animals that eat the plants, to herbivores? And the answer is yes. Otherwise, there'd be no paper, right? So what they find is that it's a complicated story. Plants emit volatile organic compounds, a whole lot of them. If we were to put a plastic bag over a plant and take it to the nearest gas chromatogram, you would find huge numbers of compounds are volatile. Okay. Some of these are noticed by aphids. We're going to talk about the aphids, which are sap suckers. Aphids come into the conversation in microbiology in many ways. But in this case, what we know is that aphids use volatile organic compounds to locate host plants. However, after they do that, plants make different kinds of volatile organic compounds which are repellent to the aphids. Okay? So this is complicated. The aphids are attracted, but then they're repelled. Notice that there's something in it for the fungi, because aphids use carbon, and fungi get carbon from their plants, and if the carbon is shunted over to the aphids, it's going to be less for the fungi to eat. So the fungi are willing partners in this, and they make for conduit of these compounds. Now, how did they study it? The authors used something very ingenious for studying this. You too read this paper, I imagine, and you may have seen that what they did is they took soil and a complicated mixture of loam and clay and silt and sand and organic material and so forth, and they inoculated this with a plantain, Plantago lanceolata, which has known, and they added a well-known arbuscular, vesicular arbuscular fungus, namely glomus. And glomalin, remember I just talked about glomalin, that protein? Right. Well, it comes from glomus. So they took the glomus, put it in the soil, and they added seedlings of the plantain, and they let them grow for 4 months. So by that time, the mycorrhizal relationship is well established. The fungus is all over the soil. And what they did is they took off get rid of the plantain, and they put beans, Shafeva. They put seedlings of beans in this place, and they put them in very funny ways. In the center is a plant. Imagine a pot with the soils, which are now full of mycorrhizal fungi. Put in the middle one of the seedlings and call it the donor, and it has aphids on it. Okay. Then put another one on by a few inches away, and that's going to receive the roots and the mycorrhizal fungi. Okay, that's just doing the same thing at a certain distance. But then put another seedling and surround it with a mesh, which is so small, half a micron in diameter. The holes are half a micron in diameter. So it must be something like a membrane filter, what we think of as a millipore or some filter of that sort. And that, of course, inhibits the contact with the hyphae and inhibits the contact with the roots. So now you have a no hyphae, no roots control. Then just because you want to differentiate between hyphae and root, you use a mesh which allows hyphal contact, but not root contact. And that is 40 micron mesh. Okay, that must look more like a real mesh. Anyhow, that allows the hyphae to go through, but not the roots. And lastly, you take that one, and then you rotate it about 90 degrees, which says that this initial hyphal contact is broken, and there's no root contact. So anyhow, they have all these experimental setups, which are very clever. I really thought it was ingenious, and I would suggest to readers, if they'd like to see a nice, simple but clever experimental design, they can go and look. Anyhow, so now they look for the aphids, and they look for what aphids do, and they do this by measuring their attraction and repulsion. And also they study something which is certainly news to me, the electrophysiology of the antennae. This is big time stuff. The antennae, you can break off the antennae and study their electrophysiology by using complicated gadgets. I won't go into that. So they study that too. Anyhow, the grand conclusion, by the way, the setup of using these plants is called a mesocosmos. Because it's 30 centimeters across. Macro would be a whole county, and micro would be much less than that. So this is meso, in between. So they did this. They studied the volatile organic compounds by covering the plants with
plastic bag.
With plastic bags, exactly, and then using that, what comes off, what is emitted in gas chromatogram. All pretty fancy stuff. Well, I don't know. It's pretty good stuff. So the result is that if the aphids, in the presence of the hyphae, the aphids are gone. But what comes instead is a parasite of the aphids. Now, you need a little bit of entomology for this. Many insects have parasites which are called parasitoids because they're parasites of parasites. And they tend to be wasps that lay their egg either on the host insect, the one that parasites, or on the eggs of it. And either way, they destroy the insect. And this is the basis for quite a bit of biological control of insects.
It's also the basis for organic gardening. A lot of the organic gardeners use these parasitoids to as organic insecticides, if you will.
Anyhow, when there are hyphal connections, the aphids are gone, the parasitoids show up. So that's actually pretty much the story. Now they analyzed and saw what is it that's involved in it. What's involved is a volatile compound that's fairly simple, namely methyl salicylate. Methyl salicylate sounds like aspirin to me. Maybe the plants use that to avoid having a headache. Anyhow, the methyl salicylates act that way, and they can act that way if you add them as pure chemicals. So you can have a control experiment where you add methyl salicylate instead of the setup that I described, just a simple setup, and sure enough, the aphids are repelled and the parasitoids are attracted. So now the question is, how do you know that all this goes through the hyphae? And the answer is that that's what the experiment is about. No hyphae, no result. Hyphae, result. So you infer this from the way you rigged the experiment. There's no direct demonstration of transfer through the hyphae. That's not an easy thing to do, and they haven't done it. So you simply infer that because the hyphae are essential, this is how it works. And there are several reasons why this makes sense. Non-mycorrhizal fungi don't do this. They don't help in this transfer. It's not diffusion because the meshes were that separate the plants were permeable to diffusion. So what is it? How are you going to transfer these compounds from one plant to another? Probably through the roots and probably through the fungi. So this is essentially the story. It's a case where the experimental design tells the story.
So can I ask you? So you put the aphids on the plant in the middle, right? Right. And then the observation is that none of the other plants that are connected by the mycorrhizae get aphids, but the ones that are not connected do get aphids on them, right?
Right. Or they have aphids. They start out with aphids.
They all started, and then they're repelled after they get the signal. That's right. And also, so the starting plant in the middle also, the aphids go away.
That's right.
Right.
Right. That's right. So
I guess the next step is to figure out what the mycorrhizae are transmitting, right?
Well, the idea is that it may be methyl salicylate.
Right. But they would have to show
it's
that.
It's the most active compound, and they can, as I say, they can repeat the experiment by adding purified methyl salicylate.
And when the aphids go, then they're replaced by parasitoids?
Yeah, but I'm sure the parasitoids don't stay around if there's nothing.
Because they starve.
Because they want to eat the aphids, right? Yeah.
It's not that they starve. They don't have a place to deposit their eggs.
Yeah, they don't make the next generation.
What attracts the parasitoids? The aphids?
Well, the chemicals as well. The chemicals too.
So the plants are making chemicals that attract the parasitoids because they'll get rid of the aphids. Is that the idea?
Right. Mm-hmm. So it's, when I was reading this, I was thinking of, the original quorum sensing concepts of how you throw out these homoserine lactones out into the environment. So you invest in the future, hoping that they come back. And so the question is, can you see fungal expression change as a function of distance so that when you have an expression change, what you're doing is it's almost akin to current of how you move an electron down a path of a wire, but rather than a path of a wire, you have these hyphae. And what we're seeing is the expression, the genetic expression triggering down the hyphal chain. And one of the things they have a time spent by the parasitoid and the aphids by minutes. And I'm just wondering how fast gene expression goes on in the hyphae to transmit these things down. And I was thinking about the first trigger is if the aphid is eating carbon, could it be as something as metaphorically simple, like the lac operon, when the cyclic AMP levels go up, you get full de-repression of an operon.
There you go.
Something simple from which you can form a hypothesis to test, because this paper is truly fascinating, and it caused me to ask myself more questions and made me want to read more about this remarkable thing. And one of the sessions you listen to at the general meeting or other places is how microbes can help with capturing carbon, and the mycorrhiza are indeed fungi, so they're microbes. And so you really begin to look at how the microbial world can help influence climate change and other things that, if we understand it better, we would know how to facilitate it.
I think you're saying what I came out with is that the paper describes a phenomenon which is believable, but that the mechanism remains to be studied.
Sure.
In other words, that's absolutely right. I think you're right about that. However, it's studyable. I mean, these mesocosms are, you can add tracers, you can add isotopes there, you can do things with it, and I imagine that there are probably in the plant. Actually, there is a literature about the transfer of compounds through mycorrhiza. I just haven't read it, but something is known about this already. Just that my knowledge of it is so small that I didn't go further. So it's a nice paper. It's a nice story.
Interesting statement in the discussion. They say it was recently found that some commercial cultivars of maize have lost their ability to produce herbivore-induced plant volatiles.
Yeah, I thought that was fascinating.
That's, I think, code for genetically modified maize.
Yeah.
The GMOs.
So the modification.
It's part of the picture. You lose something when you do that, and this is one example of something that can get lost. It could be very serious, because this could result in getting rid of a natural pest control.
Yeah, this is
by itself.
Very important if you need that for effective growth.
I'm glad you brought it up. That was really an interesting observation.
This is Michael, you're suggesting this is an unintended consequence of selective breeding, right?
Yeah. There are always unanticipated things that happen, and as you increase the height of a corn stalk, or you increase whatever you want to do, put more lysine into corn, or do whatever else they're manipulating to make corn a better molecule. And of course, there's been a lot of modification to corn to make it a better substrate for ethanol fermentation.
Right.
And so, I think, it's papers like these that, really set the ground for future studies. And this can be a transformational paper to a starting graduate student, where they just get bit by that bug that says, "Oh, what do you know about this?" and go down this path. So I found it absolutely a wonderful paper to read and just think about.
So if you grow plants hydroponically without soil, do you need mycorrhizal networks?
My goodness, that's a good question.
Well, hydroponics is extremely challenging, because you have to get the balance right on the nutrients that you're feeding the root system. And
that may say that you don't have mycorrhiza and you have a hard time because of it.
Maybe
one of the reasons for what Michael just said, that hydroponics ain't easy. So it's I don't know, but it may be. I mean, fungi grow in water, so it's not necessarily. But the thing is, one of the things that mycorrhiza do is transport water, and if you're hydroponic, you don't have that problem.
Right.
So
right.
It may not be. Maybe the mycorrhizal fungi are not seen in hydroponic plants.
Yeah. All right. Very nice. I wanted to mention an additional piece of information about volatile chemicals produced by plants. This is a very interesting story, which we covered on TWiM a long time ago, episode number 70, there is a plant virus that induces the plants to produce certain volatile compounds which attract aphids.
No kidding.
The
Oh my
aphids
goodness.
feed on the plant, and they pick up the virus and then spread it elsewhere.
The virus spreads that way.
It's brilliant. So
Huh.
the vectors are attracted to these compounds and pick up viruses. It's just amazing.
What a story.
Yeah. So
It belongs in this conversation, all right.
certainly does. Yeah, when you talked about the volatiles, it made me think of it. And so there are probably all sorts of levels of these interactions that we barely know about.
Right.
Okay, thank you, Elio. That was very nice. I like that. Our second paper is a paper published in PLOS One earlier this year. It is called Brain Microbial Populations in HIV/AIDS. Alphaproteobacteria predominate independent of host immune status. This is a paper I noticed not too long ago and I was really amazed by it and thought we have to talk about this on TWiM. And so here it is. The idea here is, everyone knows how important the microbiome is to us, and microbes are even being found in places where we thought there shouldn't be any. In healthy people, like the vascular endothelium is colonized. And in this paper, they thought, well, let's look in people that have HIV infection and who have AIDS, acquired immunodeficiency syndrome, and who are immunosuppressed. Might there be bacteria where they shouldn't be? And they thought of the brain, which, as they say, is assumed to be a sterile organ, unless you get sick or immunocompromised, in which case it may no longer be sterile. Of course, viruses can certainly get in there, and they said, well, let's look for bacteria. So that's the essence of this study. They obtained samples from people who were infected with HIV, and these individuals had various neuropathologies, toxoplasma, HIV encephalitis, herpes simplex encephalitis. These are all patients who are HIV positive. They had some surgical specimens, and then they had some controls. These are from individuals with different reasons for their problems, like cerebral infarctions, meningitis, toxoplasma, multiple sclerosis, etc.
By the way, one was interesting. It was a case of rabies.
Yes.
Yes. I don't know where they found that.
Yes, thanks for bringing that up. So these are all autopsy-derived material, okay? These people have died for various reasons. So they have material, and then and there's quite a few of them, about 8, 15, 20 or so total specimens. And they extract RNA from them and do some sequencing to try and identify whether there are any bacteria in these sequences. So they do total sequencing and then use computational methods to look for the bacterial or viral sequences. They did both. And amazingly, they find a lot of bacteria and phage-derived sequences in these samples. 173 different phage and bacterial-derived samples, and mostly alpha... Yes, go ahead.
I'm sorry. One thing, you said they were all autopsy material. I don't think that's quite right. Some of these were just done in surgery, and I think it makes a big difference. So I want to make that point. That
Let's see. Total RNA.
The epilepsy resections.
Right.
Yeah. So the HIV/AIDS,
biopsies.
the HIV/AIDS were autopsy, and the others were not. Yeah, all. Okay, sorry about that. Yeah, I was saying that as I was reading it. So some of these people are still alive. All right, so they find lots of bacterial sequences, a lot of alphaproteobacter sequence tags, and in fact, every sample, not just HIV/AIDS samples, but all of them have sequences from alphaproteobacteria.
That just blows my mind.
Isn't it amazing?
That's not what I learned when I was young.
Yeah.
Not supposed to. That's not supposed to happen.
And as you'll see, they verify that. Lots of bacteriophages, which are specific for proteobacteria. And the one patient with rabies, they found, guess what?
Rabies?
Rabies virus. They got 20,000 rabies virus sequences spanning the entire viral genome. They got the whole virus out of this one person. There's a lot of rabies virus in the CNS. And they also found some other viruses as well, not as consistently present as the alphaproteobacteria site. A bunch of herpes viruses, basically. But again, not as often as the bacteria, just in a few, and sometimes not any of the specimens. So the bacteria, the alphaproteobacteria, are found everywhere.
But one thing you should point out to those listeners that aren't familiar with what alphaproteobacter are, this group of bacteria mostly comprise phototrophic genera. And the last time I looked, even though my head is a little hollow, there's not much sunlight in a brain. And
unless you have a good idea.
And
but there are plenty of alphas which are not.
Well, that's true, but
synthesizers. So I don't know about that.
And they also go after C1 compounds, and the brain is very glucose laden. So it's interesting that the alpha proteobacter have several genera metabolizing C1 compounds, but they also include in that group, our friends the wall-less bacteria like Rickettsia.
Mm-hmm.
So
not wall-less. Wait a minute. You think of mycoplasma?
Oh yeah, Rickettsia.
You know why I say that? Let me tell you why I say that. Because 1 million years ago, I worked on Rickettsia, and we discovered that Rickettsia have cell walls.
They got a little peptidoglycan. That is true.
Not a little. They have a fair amount. So, anyhow, that's why I took the liberty of interrupting. Sorry.
Sure, no, it's fine. We got to make these corrections. Otherwise,
we get letters.
So the alpha proteobacteria are in, again, all of these specimens at high numbers. Some actinobacteria, but not as many sequences as the alpha proteo, and none from the surgical samples. Beta proteobacteria, even less, and even in the same very low gamma proteobacteria. They say that there is, for example, in terms of phyla, no firmicutes, which you find elsewhere in the body, right?
Especially in the gut.
Like Clostridia.
Yeah, and then actinobacteria were minimal, as I said. Okay, they also look at some host genes, and they find that genes in HIV, the specimens from HIV patients that are involved in immune responses are elevated, as you might guess, because these individuals have an ongoing virus infection, but not as much as, or less so in patients without infection, the surgical specimens. You don't see elevations of these immune response genes. Okay, the next part, which I think is interesting, is they say, can we see bacteria or bacterial products in sections of brain from these specimens? So they took the material from either the HIV patients or the other conditions, and they made sections, and they stained it for various antigens. And they look for peptidoglycan, this bacterial wall constituent. And they can find it in both patient sets, again, in the HIV set and in the other set as well. They can stain, and they can see small particulate staining regions corresponding to something reacting with antibody to peptidoglycan. It's in the white matter. And they can they also see it in blood vessels. And by using markers for different cell types, they can find it in astrocytes. This is within the astrocyte, microglia, and macrophages. And again, this is in both the HIV and the non-HIV samples. So I don't know if you gentlemen have looked at these images. They're
incredible.
Do they look like bacteria?
Well,
it's hard to say, right?
Some have the right shape. I've never done enough histology to know how histologists. I think you have to look at lots and lots of images to infer some of these things.
There's certainly discrete staining, right?
They're discrete staining. This is true, and it's immunohistochemistry.
Yeah. I mean, what's interesting to me is that often they find them within cells, right? It's not
that's not surprising, considering that a lot of bacteria that infect humans and other places are facultative intracellular parasites, and monocytes and macrophages pick them up, and the best example is, of course, listeria. Mm-hmm. It can get picked up and be carried around by our cells, and we allow macrophages and we allow monocytes to get into our brain, and once they get in, listeria is a good example. That's something that can get out.
Yeah.
And so, I think this is an instance of where the microbes don't really set up an abscess like the traditional infectious process, which we associated with, common things like the best example that I come up with always is a boil, because people understand pimples or, the classic abscess.
Yeah.
But occasionally a brain can develop an abscess.
Yeah, so, these guys, this is the big difference here, isn't it? They don't.
Yeah. No.
It is totally counter everything I ever learned. I mean, I really did think that if bacteria are found in the brain, it's because they cause symptoms.
Right.
Here they don't. It's amazing. It's totally mind-blowing.
It is.
Well, when you look at Legionella and Haemophila, we know that in the late stages of chronic Legionella, you can actually recover Legionella antigens from the brain. That observation was made when I was a graduate student in the early 80s. And so consequently, in Legionella and the issues associated with Legionella and the subsequent sequelae of that chronic infection can cause neurological symptoms. So,
but that's right. They cause neurological symptoms. That's the whole point. I know that bacteria can infect the brain. The question is, can they infect the brain and not cause symptoms? And this is what they're saying.
Yeah,
this is why this is so amazing. It's totally. I know bacteria can enter the brain. They do it all the time, but they cause symptoms.
Yeah. And here they're not. So
the implication is that the implication is that they're probably in all of us, right?
Yeah.
That's mind-blowing.
Literally.
Was that unintended?
Who knows? With me, you never know.
And, since I have the great pleasure of teaching dental students, and I talk a lot about the microbes of the oral cavity and how they can actually penetrate. And so I'm wondering, as periodontal disease is increasing in the country for a wide variety of reasons, I'm wondering as your dental condition deteriorates, whether or not the concentration of bacteria in the brain actually increases.
Tell me, are those diseases caused by alphaproteobacteria?
Well, the oral cavity has about 1,000 different species. There's there are lots of gram negatives in the oral cavity. Most are strict anaerobes, and there's quite a few that have been uncharacterized. And so alphaproteobacteria are in the oral cavity. I just don't recall at the top of my head what percentage they represent of I have to go look at the microbiome literature to see what percentage alphaproteobacteria are associated with periodontal disease. I think they've gotten the deep sequencing done of the periodontum, and so I'll take a look at that. That was one of the things I didn't get a chance to check.
By the way, I was going to ask you, Michael, would you explain why deep sequencing? First of all, you may want to tell it. Not every reader knows what that means. But can you explain why they insisted on deep sequencing and why it makes a difference?
It's the size of the DNA that they actually recovered, because their fragment size was so small. They want to sequence as much to get a representative sample of who's there.
Doesn't the deep sequencing refer to the number of rounds of
Right.
Yes.
the number of cycles?
Yes,
It's the number of,
Why is this important here? What?
Well, it's an unbiased approach. You sequence everything, and since
I see. That's what it says.
since there are things present at very small
Okay,
quantities,
you're grabbing the rare.
yeah, you have to do massively parallel deep sequencing, as they call it, so that you can pick up the low abundance as well as the high abundance stuff. Yeah,
That's right. It's designed for low abundance.
yeah,
and that's what they're effectively looking for, to figure out who's there, to get at your principal point, Elio, that these are bacteria that are in the brain that are considered normal flora. Because I think that's effectively the take-home message. The brain have normal flora.
right.
And if they have normal flora, is this area of the human body, is this microbiome relevant to mental health?
Exactly.
Hey, maybe it helps us develop neuronal connections. Who knows?
Yeah. Well, it goes back to our mycorrhizal discussion.
Yeah. Maybe
Is that the connection between these two papers?
Sure, there you go, Elio. You knew that, didn't you?
Well,
I was wondering.
You look at volatile organics, and we know the bacteria in our guts make these low molecular weight compounds that can move via our circulatory system. So the question is whether or not these homoserine lactones, because gram negatives use homoserine lactones, whether or not these things are reacting and stimulating various aspects of development of our brain.
Sure.
The president has said this is, a priority for the NIH is the brain. And if there is truly a brain microbiome, as this paper may actually suggest, then the question is, what role does it play in base intelligence or mental health or
Huge.
attention?
Yeah.
Yes, a simple question: attention deficit disorder.
Yeah.
You could phrase the hypothesis: is the increase in attention deficit disorder a consequence of the elaborate use of antimicrobials, because many of our children, or this generation that has ADD, has been in daycare and they have been overly prescribed antimicrobials so that they can get out of fever jail.
Right.
Wow.
They make the point that these bacteria, there's a particular family, Sphingomonidaceae.
Yeah.
They synthesize and incorporate sphingolipids in their outer membranes, which of course are important components of the brain too.
And sphingosine kinase is the wonder enzyme in cancer therapy and all of these things. And so,
tell us about that. What do you mean?
Well, this whole emerging field of lipidomics is circulating around sphingolipids and sphingokinases, and they're looking at the role that these molecules play in the development and control and actually in therapy of cancer. We have a few groups on my campus that are actively working on this. Perhaps the most famous that has since left our institution and moved up to Stony Brook is Dr. Yusuf Hanoun, who's one of the world's foremost experts on lipidomics and the whole issue of sphingosine and sphingokinases and their role in cancer. Wow. And I would commend you to take a look at some of his papers specific to cancer. But again, finding a microbe that has got this sphingo connection was really extremely interesting, and it led me to really begin to warm to the idea that there may indeed be a microbiome for brain tissue.
Could be the name of this episode, right, Michael?
Yeah.
The brain microbiome with a question mark after it.
Yeah.
Very good.
Let me finish up the experiments here. There are a couple more I wanted to tell everyone. First, they confirm this deep sequencing by PCR. They amplify ribosomal RNA, 16S ribosomal RNA, and they confirm the identification of the bacteria this way. And they go to very great lengths to make sure there is no contamination.
Right.
All right. Very carefully done. You can read in the paper if you'd like. And they also, they look for specific bacterial transcripts, and they find them. The one that they look for is the GROW-EL transcript. They find that.
One of the most abundant. It's one of the most abundant proteins in bacteria. So,
and here's an interesting thing. They do not find these bacterial sequences in the brains of cats or mice that are maintained in pathogen-free environments. So, who knows if these animals are normal because they lack these bacteria? It's hard to tell, right? You can't talk to them. But it may be that they have altered behavior because they lack these bacteria. It's interesting.
And the neuroscientists are actually using these animals in germ-free facilities, and they're doing behavioral studies on these.
Maybe all screwed up.
Well, no, I'm just saying.
This is very interesting. All right, and here's the last one. That's very cool. They try and transmit these bacteria to other animals because Elio would say, you've got sequences, but who knows if they are living bacteria?
Right. I would say that.
Well, that's his channeling of Norm Pace.
When I was reading this paper, I was thinking of it. I said Elio's going to say that. So what they do is they make homogenates of these brain samples. They either treat them with heat or do not treat them with heat, and then they implant them into a very specific place in mice. And these are immunocompromised mice. They're called RAG knockouts. RAG is a gene that is important for recombination in the development of the immune response. And then they keep the mice for 7 weeks, and then they extract RNA from their brains. And what do you think? In the mice that received unheated brain homogenates, they find sequences of the same bacteria that were detected in the human specimens at a 40% abundance of the original implanted homogenate. And of course, if you heat it, then they don't get any bacterial sequences in them. So what do you think of that, guys? Is that a good experiment?
That fulfills Koch's postulates, in my view.
Well, yeah. I still would like to see a colony on agar, but that's okay. I mean, these are bugs that grow pretty well on agar, and so
yeah, it's true.
They are, what's going on? Are they viable but not cultivable? Whatever that means, I never was sure I understood that, but or is something else going on? So it's certainly. I wouldn't say this is Koch's postulates yet. Close.
Yeah. But it's better than not doing any transfer at all, right?
Yeah.
Because then
I'm not making a case against it. I'm just saying there's more to this, and we have to know more.
I'm sure they and others will be trying to culture some of these and and prove all this, because this is potentially huge, right?
It is huge.
Or could, of course, it could be all wrong, but we'll see.
Well, I give them credit for doing the conformational cloning experiments of cloning the genes out into bacteria and doing the full 16S sequence.
Yeah.
Because that at least shows you that the deep sequencers did actually work with bacteria and not just PCR reagents. Oftentimes, if you go into one of these microbiome labs, they just have plenty of stool samples or plenty of other samples, and they don't culture anything ever. So I give them credit for at least working with microbes.
Yep, got a good point there.
So lots of interesting questions raised by this, and of course, one we just talked about: are these beneficial? This would be very, very cool. I bet they are. If they're there for a reason, right?
Well, the brain is immunoprivileged, so once they get in there, and the other question that I would wish there was some really good way of doing this, but I don't think the sample size is big enough, is ask how many are there, because what's controlling their number?
Yeah, I was going to bring that up. There's no quantitation here. It's yes or no. You find them or you don't find them.
Well, that's the fundamental problem.
Doesn't tell you how many.
Well, you can tell by the how many sequences you get roughly. You can compare specimens.
Pretty rough.
But yeah, it doesn't tell you the number.
What's keeping their number at a subclinical level where there's actually disease? Because it all goes back to what Elio brought up earlier. Yes, I believe bacteria can get into the brain because that's known since the beginning of time in terms of infection and disease. But the question really is what's controlling their number and their distribution? And do they like certain hemispheres of the brain over others? Do they like the cerebellum? Do they like the brainstem? You look at these things, and this is how we're going to figure out if there is indeed this microbiome is doing good work, or is this just mere a consequence of, as we age, the bacteria begin to leach in, and the immune surveillance system of monocytes and macrophages keep them under control in the brain as those surveillance cells effectively do general housekeeping.
Yeah.
And the real experiment that needs to be done is on the population of Alzheimer's folks. And as they donate their brains on autopsy, to ask the question, who's there in the Alzheimer's population? Because what's actually forming those tangles that result in the development of Alzheimer's?
Yeah.
Very good point.
Michael, can you, are there non-invasive ways to look at this?
Well, the new MRIs with contrast, I guess, are one way of doing it, but I don't think they have sufficient resolution to see microbes.
Yeah.
And that's why I wanted to know about number, and that's the other thing I'm going to torture my radiologist friends when I see them in the cafeteria, is what's the resolution of an MRI? I know what the resolution of a light microscope is, but what is the resolution of an MRI? Will I be able to see bacteria inside brain cells?
Maybe you could give a dye that would be taken up into the bacteria also to help, right?
Sure. Yeah.
Something that wouldn't be toxic, obviously.
Something that wouldn't be toxic.
Because it would really be interesting to look with development, right? As over time, and see if there's change or what,
Well, it
cool.
takes us back to that TWiM episode we did last year on the development of the autistic children, and we looked at when the group at Columbia, your colleagues looked at the populations, the ones with stomach issues that didn't have autism and the one did, and they found the Sutterella species, which is a derivative of Bacteroides, which is Prevotella. And so the question then is, were these things present or absent in the brain? Do they have to be there at an appropriate period of time? And temporally, does the microbiome of the brain change?
Right.
And so, again, this paper forces me to ask more questions than it answered. That's what science is about.
Good papers don't answer questions, they make new ones.
That's right.
All right. Anyway, that's very cool. Check it out. We'll put the link in the show notes. And I think we'll be hearing more about this in coming years.
No doubt.
Let me read a couple of emails here. We were kind of backed up because we've been traveling. The first one is from Daniel, who writes, Dear Vincent and his fellow TWIMsters, my name is Daniel. I am a first-year graduate student at Michigan State. I would like to thank you all for making a great podcast. I recently caught up to the most current episodes by having my own TWiM marathon during my 6-hour drive home to Wisconsin over the holiday break. I'm not sure where to begin explaining how much I value this podcast. As someone who cares for outreach, education, research, and in general having a good time with science, you folks are first rate. This series has been accessible all the way through for grandparents to grad students. When things get maybe a little too verbose, you have done well to bring it back down. But you also make it just intriguing enough to hold the attention of the grad student in me. What
does he mean too verbose? Come on!
He's probably talking about me.
That's how all of us are probably guilty. I have even been known to actually follow along in the articles. Wow, that's impressive. And of course, I have had a great time learning and exercising my mind while listening in. You guys went even further to impress me with the discussion of Joe Handelsman's research last time. It gave me lots to think about for the future. Please keep up the great show. I also wanted to share a paper with you all that I thought about after listening to TWiM 47, Resistance on the Surface. Michael said something that made me remember it during his discussion of the paper in regard to conjugation. He said it's having sex wicked fast, and you guys went on to exclaim about how fast conjugation was happening. In this science paper by Babic et al. In 2008, they are using a very beautifully designed experiment to visualize DNA transfer by conjugation. And I agree, it's wicked fast. I suggest the supplemental videos. The experiment involves a recipient strain that is DAM- or defective for DNA methylation by DAM methylase, and which has a SEC-A YFP fusion. SEC-A being, correct me if I'm wrong, the protein that keeps replication from occurring when hemimethylated DNA is present in the cell, effectively keeping multiple rounds of replication from occurring at once, since newly replicated DNA has not yet had the time to be methylated on the new strand. The result of this recipient cell is no DNA methylation or SEC-A YFP binding and diffuse fluorescence. The donor cell, however, has DAM methylase and will donate methylated DNA during conjugation. When this happens, the donor DNA will become hemimethylated upon replication, allowing SEC-A YFP to bind and show a localized fluorescence to that DNA. This means we can watch it happen at a single cell level. They do show a rather surprisingly fast transfer of DNA, as well as make other neat conclusions in the paper, which include the use of a red fluorescent gene on the donor DNA to measure expression of conjugated DNA, but this email is long enough already. It's not a bad read, and I hope my explanation was TWiMable enough.
Well, I read the paper carefully when it appeared, and I've sort of forgotten the details, but it certainly is a beautiful paper, and I think he's right in calling attention to it. Thank you very much, Daniel.
I'm just pulling it up here. It was published in March 2008. Direct visualization of horizontal gene transfer. There you go. Thank you.
It's a very, very, and the videos, if you have a spare couple of minutes at lunch, go online and check out the videos. It's good lunchtime watching.
Thank you for providing a wonderful supplement to my microbiology education, and Happy New Year. Wow, this email has been sitting around for a while, I guess. It's New Year's 6 months ago. Sorry, Daniel, but thank you for that. That's great. Robert writes, I am listening to episode 49 and heard the mention of biodynamic vineyards. Biodynamic farming is similar to organic farming, with the addition of such things as burying dung in the horn of a bull and sprinkling magical powders around the farm. Take a look. It's quite entertaining. Total nonsense. Do you know that, Michael?
No, I didn't.
What are we talking about, biodynamic? That was the vineyard microbiome.
It was the vineyard microbiome where they, had a normal developed farm, and then they had the organic farm and the one in between. And, the thing I most remember about that is how they went about sampling the vineyard to show that they actually had a true representation of the microbiome associated with the grapes. And but this one, which he's writing, I have to go and hunt up and see what he's talking about.
I'm looking at this on Wikipedia. It's very interesting. For a vineyard to be considered biodynamic, the wine grower must use the 9 biodynamic preparations described in 1924. These are made from cow manure, quartz, and 7 medicinal plants. And he's right. You put cow manure in corn horns and bury it. You put ground quartz in the cow horns and bury it over the summer, et cetera, et cetera. Who knows what this does, right? He may be right. Anyway, that's certainly new to me. Thank you for that, Robert. The next one is from Todd, and he writes, it's true, and he gives us a link to an article on why some Civil War soldiers glowed in the dark.
Oh, yeah.
You guys know this story?
Yeah,
No.
I do. It's
This is
about
one thing I don't know.
microbiome. Let's see if I have the name right. Cousin of the bug that kills nematodes.
Photorhabdus.
Photorhabdus.
Right.
Exactly. And it does cause, it glows, it's bioluminescent, and it is found in soils, I believe. Yeah, of course. And it was common in trench warfare in the First World War, where wounds got routinely contaminated with soils. And this bug is fairly benign, so that when people saw a limb glowing, that was supposed to be a good prognosis. Whereas if they didn't, it was supposed to be deadly because something else was infecting. So that's an interesting story.
Yeah, so they
I
grow
don't remember
in,
much.
They grow in nematodes in the soil, apparently.
That's right.
And they're released when the nematodes vomit them.
Right.
And the idea here the story was that these Civil War soldiers were glowing in the dark one night during a battle. So what they think happened is the bacteria colonized their wounds, and that's what was doing that. But
as I say, it was supposed to be a good prognosis.
Oh yeah.
And I think it's appropriate that this letter is associated with this episode because the photorhabdus luminescens sends an invitation to the party, and I'm reading from the link that he sent just before they got back in their nematode taxi. P. luminescens were at critical mass in the insect corpse, and scientists think that they may be many glowing bacteria attract other insects to make the body and make the nematodes transition to a new host much easier.
Nice.
So this is like the mycelia with the aphids.
It's a cool article. I suggest everyone check it out. Trod also finishes up by saying, I love your podcast, and in the future, when I write a longer letter, I'll explain why I love the podcast. Okay, we'll wait for that, Todd. Next one's from Eric, who writes, long-time listener, second-time emailer, ID doc. Mostly I treat HIV patients and hospital-acquired infections. You've talked a lot about copper as an antimicrobial and that ancient civilizations might have used it to reduce war wound infections. I'm not aware of its use in modern wound care. However, silver-based products are used a lot in wound care and on catheters, etc. I would be interested in understanding the antimicrobial properties of silver and why copper is not used topically if it is so effective at killing bacteria. Thanks. That's for you, Michael.
Question.
Well, the simple answer is copper is also toxic to eukaryotic cells, which is why we don't bring them in direct contact with living cells. Our surface skin layer is effectively dead, so the copper is not going to have an effect. Silver, in contrast, works principally through leaching, and it's the silver ion, Ag+, reacting with the free self-hydro groups that renders them inactive and activating the protein. And silver is effective simply because it leaches. And the silver catheters, typically, when you place that catheter inside the body at the local site of catheter placement, you can actually have a blood level of 250 micromolar silver ion at that location. And of course, as some folks may know, those of you who ingest colloidal silver, you can have a very bad side effect where the silver accumulates and goes up into your skin itself, being distributed via your microcapillary system. And then the silver, because we all know about photography, is photoreactive, and you'll actually turn the color of Smurf. You'll actually become bright blue. And that, unfortunately, is a precipitation reaction, just like black and white photography. And it doesn't go away. And so silver has been used a lot in bandages and things that come in direct contact with mucous membranes and tissues, simply because it's a leaching-based technology. It's generally coupled with EDTA, and it trickles out at a defined rate so that you achieve a local level to inhibit the microbial growth.
Isn't it nice to have an expert on board?
That's right. It's great.
It's good.
Thank you, Michael.
We try.
The next one is from Jim. I thought Michael might like this reference. It's to a Canadian podcast called Quirks and Quarks, and he provides a link. In this episode, they discuss a paper by Jane Danska talking about diabetes in non-obese mice. The outcome of diabetes can be changed by changing the microbiome in young mice. It reminded me of Michael's thinking on how the microbiome might come into play in the future. I found it very interesting after hearing the TWiM episode. So I don't know if I guess Michael Jim is referring to our discussion of the Danska paper in TWiM 54.
I think so.
And so now you can hear Jane Danska talking about it on this podcast. So
I'll look forward to that.
Thank you for that, Jim. Just two more. The next one is from Justin, who writes, "Hello to the TWiM team. I've written to TWiV, but this is my first letter to TWiM. I was recently watching a documentary about how beer was the driving force behind beginning agriculture and numerous other massive human accomplishments, and the documentary mentioned skeletons from 500 BCE containing tetracycline. My first reaction to this was, where is my computer? I have to find this article. Well, I did find that original discovery in the late 1980s was met with extreme skepticism, not surprisingly.
Why?
But then I also found an article from 2010 with more data to back up the claim. Now, I am not one to interpret mass spec data very well, but would love to hear what the TWiM teams think of it." He sends a link to a paper in the American Journal of Physical Anthropology entitled Mass Spectroscopic Characterization of Tetracycline in the Skeletal Remains of an Ancient Population from Sudanese Nubia 350 to 550 CE.
We better go and read it, I guess.
Well,
I've heard of that paper, but I haven't read it.
The thing I remember about tetracyclines is there are a series of hydrocarbon rings with various side chains that give it its various level of activity. And it's in a class of polyketides. And I know there are bacteria out there that make polyketides, so it could be something very similar to the earlier letter where we had the glow-in-the-dark bacteria from the nematodes. We may have just had microbes from the soil that were making polyketides that were making tetracycline-like molecules.
The mass spec would pick up, I missed. Well, it could be.
So
in the database.
So you don't think these individuals ate streptomyces that make tetracyclines?
They could have done that.
That's the alternative.
Yeah.
They could have done that too.
I mean,
but I know about the polyketides because one of the guys in the School of Pharmacy was working on polyketides, and I remember going to a few of his seminars, looking at the structure and talking about how you make substitute side chains, and if you really want a organic chemistry review. Look at things like tetracyclines and macrolides and then ask yourself the question, how did the bacteria ever get through organic chemistry? Because the reactions that these microbes do are, the graduate or the undergraduate in organic chemistry's worst nightmare for a final exam synthesis, because there's no way you could ever design a synthesis reaction in order to make these derivatives. Yet the microbes do it quite well, and it's simply because of how they make everything in the cytoplasm, and they compartmentalize things by passing intermediates from enzyme to enzyme to enzyme in a very small volume. There's an advantage to being only having 10 to the minus 15 liters to do your reaction chemistry in.
Well, this paper is mostly mass spec, and it's clear that they're identifying tetracycline derivatives, but what they say in the discussion is this population of individuals were very good at the science of fermentation. They made gruels or beers, and they say now they could have produced mixtures with streptomyces or other species. And maybe they did this on purpose. Maybe they found it had beneficial effects, right? Who knows?
Well, just remember how we made penicillin in the Second World War. I think it was rotten cantaloupes.
Yeah, so I'm not sure that we'll ever get to the bottom of this, but it's not unlikely, it seems to me.
Right.
The last one is from Robin, who writes about Alaric the Great. Now, we must have...
Goodness.
Did we talk about Alaric on one of ours?
I don't remember Alaric.
Alaric the Great.
You must have, because he says...
Goth, was he?
Yeah, he says one...
Visigoth, yes.
Alaric the First was Gothic, king of the Visigoths from 395 to 410, the most famous for his sack of Rome in 410, which marked a decisive event in the decline of the Roman Empire. And Robin notes, one would expect that Italians would be familiar with Alaric the Great. And that is a jab at you and I, Elio.
Right.
Or maybe you weren't on the show, but I guess I just expressed my ignorance of Alaric, and so here we have what he did. So thank you for that, Robin. And that will do it for today's email. We have plenty more left, and we'll save them for next time. You will be able to find this episode at iTunes and at microbeworld.org/TWiM. If you like TWiM, a good thing you can do for us is to head on over to iTunes and give the podcast a rating. That helps to keep us visible so more people can learn about, things like bacteria in your brain, the brain microbiome. And we do like getting your questions and comments. You can send them to twim@twiv.tv. Elio Schaechter can be found at Small Things Considered. You should check out that blog. Thanks for joining us today, Elio.
My pleasure, as always.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
Thanks for coming to the ASM during your meeting. I appreciate it.
Oh, it was a great joy. I got to play with the real Tinker Toys.
Just make sure you turn the lights off.
I'll leave everything as it was.
Turn the lights off when you leave. I'm Vincent Racaniello, and you can find me at virology.ws. I would like to thank the American Society for Microbiology for supporting TWiM, and particularly Chris Condine and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.